Dr. Pade is a research scientist in Certara's modeling and simulation group. He received his doctorate in pharmacokinetics from the University of Texas at Austin. His major interests include developing animal PBPK models for preclinical species and bariatric surgery models to evaluate the impact of weight loss surgery on pharmacokinetics. Devin, welcome to today's event. I'll now turn it over to him to begin the presentation.
Thanks, Susan. Welcome to the webinar, everybody. Good evening, good afternoon, good morning, depending on what your location is. The webinar today is titled Precision Dosing Using PBPK Modeling, and we'll be looking into how PBPK modeling can be used to optimize doses in specific populations and how it is a part of the precision medicine overall scenario. So before I start my presentation, I would just like to do a quick poll on the familiarity of the audience with respect to PBPK modeling. So I have a simple question here. Can PBPK modeling and simulation help with precision dosing? I would like everybody to answer either yes, no, or I don't know. Okay, so we have a 66.7% of our audience think that it's yes, and 33.3% think that they don't know. So we'll be aiming towards, after the presentation, we'll be asking the same question and we'll see how the statistics change. So this is a brief outline of the presentation where we'll be discussing about the Precision Medicine Initiative. Then we will move on to what is precision dosing and how can PBPK models help with precision dosing. In that, we'll be looking into what kind of population are we looking at? Is it what kind of the right patient are we looking at? And then what kind of dose optimization is required in terms of precision dosing? And how can PBPK models help with generating the right patient and in optimizing the right dose? We'll be also looking into some brief examples with respect to the current scenario and how PBPK models have helped in optimizing the right dose for the right population, and also looking into future applications in terms of precision medicine and precision dosing. The Precision Medicine Initiative was passed on by President Barack Obama in early 2015, and what it dealt with was basically that the Precision Medicine Initiative will target to deliver the right treatment to the right patient at the right time. In terms of this, the initiative basically aimed at assigning $215 million for the fiscal year 2016, from which $130 million would be dedicated towards generating a large national cohort program in which it will involve 1 million participants. And this large cohort program will basically deal with generating a large amount of data with respect to the population. And $70 million have been dedicated towards the National Cancer Institute towards research in cancer genomics. So in terms of precision dosing, when we are thinking about precision dosing, it's basically an integral part of the Precision Medicine Initiative, because without having the right kind of patients and without knowing the details of the right patient population, you cannot arrive at a precise dose. And modeling and simulation techniques or pharmacometrics, whether it is top-down or bottom-up, the modeling and simulation technique used is a very crucial link between deciding on the precise dose and delivering it to the right patient population. So with regards to the modeling and simulation technique, these techniques, whether top-down or bottom-up, they have been very, very successful in drug research and development, but they have not yet been very successful at the point of treatment or in the clinic. So though the precision medicine initiative was rolled out in early 2015, the FDA Center for Drug Evaluation and Research has already been evaluating drugs based on personalized medicine treatment. So if you look here, in 2014, out of 41 novel new drugs that were approved by the FDA, 9 or 22% of them were classified as personalized medicines. And this trend continued on in 2015 when the FDA approved 45 novel new drugs, and out of those, 13 or 28% were classified as personalized medicines. The classification here was basically was done by the Personalized Medicine Coalition, and the classification was based on the labeling information that was provided in the drug labels. And this information was basically based on biological markers which led to procedures of specific treatment to individuals or patients. Now, biological markers could be anything depending on the enzymatic status or the transporter status of the patients. So, as you can see here, in 2015, there was Aristara or aripiprazole, which was an injectable formulation, and Rexulti, which is brexpiprazole, and Serdelga, which is eliglustat, which was approved in 2014. Now, the treatments for these were basically influenced by the CYP2D6 status of the patients. Similarly, we have Biliodac, or the FDA-approved Biliodac or Belinostat in 2014, where the treatment was influenced by the UGT1A1 status in the patients. And this data is basically from the Personalized Medicine Coalition report, which was published by the FDA in early 2015 and 2016 for the years 2014 and 2015. So when we are looking at the use of PBPK models to decide on precise dosing, so first we look at what is a whole body PBPK model. So a whole body PBPK model is a model that basically takes into account most of the tissues in a species body and accounts for the physiological and anatomical composition of the body. So basically, a whole body PBPK model will track the behavior or the course of the drug from the site of administration to the site of effect, or to the site of tissue where the effect is needed. So one of the main differences between a PBPK or a bottom-up model versus a top-down model is that in bottom-up models or in whole PBPK models, you do not require previous clinical data on which your model is based. So without any previous clinical data, whole body PBPK model can actually predict or generate plasma concentration time profiles for a particular drug in the plasma, as well as they can generate a drug concentration time profiles in various tissues that the whole body PBPK model is comprised of. So each of these compartments that you see in the whole body PBPK model, they are relevant towards the volume and the blood flow rates of the blood, as well as the various composition of the tissues. So, for example, when you see some of the compartments here, which are with a bold border, you can see that these are some of the main sites of drug disposition, such as the liver or the kidney or the brain or the gut. So when we are looking at a whole body PBPK model, you kind of have a systems approach, wherein you have the systems data, where you're talking about the details of the population that you are studying. So you have the age, the weight of the population, the tissue volumes that make up your model, the composition of the tissues that make up the model, the blood flow rates, which is again summed up by the cardiac output, and then the blood flows to each of the tissues, the abundance of plasma proteins, etc. Then you have the other component, which is the drug data. So when you have the drug data, you're looking at the characteristics of the drug, such as the molecular weight, the partition coefficient, the ionization or the pKa, how does the drug bind to the plasma proteins, what is the blood to plasma ratio, what kind of characteristics does the drug have with respect to in vitro metabolism, with respect to permeability, solubility, etc. And then the third component of the model is the trial design, wherein you are looking at what kind of dose is being administered, what is the route of administration of the dose, are there any co-administered drugs along with the dose, and again, what kind of a population that you are dealing with. And all these parameters basically are taken together and using mechanistic IVIVE, which stands for in vitro, in vivo extrapolation, and your whole PBPK model, you can actually predict the plasma or the tissue drug concentration time profiles in the population of interest. So when you're handling these all parameters separately, so for example, if you're studying a certain drug X, then you can look at the effect of that drug X or the disposition of that drug X as you can see in the systems data here, we have a representation of the cirrhosis population. So, and then you can actually, keeping these three aspects independent, you can change each of these aspects. So basically, you can shift from a cirrhosis population and see the effect of the same drug in an obese population, or you can change the drug and see the effect of it in another population. Similarly, we have different kinds of populations. So you can change the population libraries from a ethnic population to a geriatric population or a pediatric population. And at the same time, you can also change the drug and see the effect in each of these permutations and combinations. So in the end, what you have is that you're trying to figure out what is the right drug for the right patient and what is the right dose at the right time. So this is how basically we are looking at how PBPK models can help with precise dosing, because these are the three main aspects: delivering the right dose to the right patient at the right time. And with the design of the PBPK platform, the way it is, you can handle these very independently. So we'll look at the first aspect, that is replicating the right patient. So in terms of, usually when you have a clinical trial, sometimes it's in a healthy volunteer, but when you're shifting from a healthy volunteer to a disease state or a ethnically different population, there are many parameters, or there are many system parameters that would change. So in order to have an idea of the precise dose to a particular patient or to a particular population, you need to know in depth the characteristics of the specific population. So in SimSip, when we are generating virtual individuals, so when you're talking about bottom-up modeling, you're basically, what you're doing is you're actually generating virtual individuals from bottom-up using the basic characteristics of the population. So this is basically a kind of a map of how in SimSip we generate virtual individuals. So when you're specifying a trial design, or when you're specifying the characteristics of a population, you first begin with the distribution of the sex and the age of the population, and depending on the range and depending on the characteristics of these two parameters, you can see that there is a direct correlation of the sex and age to the height and weight and to the body surface area. And hence, body surface area is again then connected to heart volume, liver volume, cardiac output, and all the other characteristics of the tissues that make up a particular species. So in that, if you add other covariates such as ethnicity and disease, so these are also again taken into account, and these basically govern the covariates that basically go into generating your population. So when you are looking into generating these populations, you are actually looking at the distribution of the tissues for that particular age range or for that particular gender or sex. So you are looking at when you're generating these virtual individuals, we are actually picking up the size of the lung for that particular age range, because that age range is connected to body surface area, and body surface area is connected to all the other covariate parameters. So depending on those inputs, basically you are selecting the size of the tissues based on these distributions. So when you're generating virtual individuals, basically you're generating all these individuals based on picking up the particular tissue parameters from this distribution. And so what you have is that you have a basic prime difference between the sampling, which we call as random Monte Carlo sampling, versus correlated Monte Carlo sampling. So in random Monte Carlo sampling, if you don't take into account these covariates, what you're looking at is that you are generating an individual with all the body tissues and the parameters and etc. But in the end, when you have your virtual individuals, what would happen is that you might probably generate an individual with a disproportionate tissue parameters, such as an individual having an abnormally large liver or extremely small gut or something like that, because it is not correlated. But when you have a correlated Monte Carlo sampling, what you're doing is that because you're picking up the tissue parameters from that distribution that has been embedded into the PBPK platform, you are generating the individuals of the right size. And hence, when you're generating individuals of the right size, and because of this distribution of these tissue parameters, when you're generating a healthy individual, you are generating a proper healthy individual, or when you're generating a pediatric individual, you're generating a proper pediatric or a diseased individual. Hence, that's the main difference between random Monte Carlo versus correlated Monte Carlo sampling. So just imagine now, if we have these kind of distributions for each of the diseased populations or each of the ethnic populations that are embedded in SimSip, what you're looking at is that you are actually generating meaningful individuals for a certain patient population. So so these are some of the characteristics of some disease populations that we have in SimSip. So I'll be just going through a couple of these disease populations. And as you can see here, when you're looking at the SimSip liver cirrhosis population, based on the demographics, you can see that most of the liver cirrhotic population is between the age of 40 to 60 years. So when you generate a cirrhotic population in SimSip, you are generating actually individuals which are between the age of 40 to 60, because that's what the data or the literature suggests. Similarly, when you look at the severity of liver cirrhosis, because liver cirrhosis is categorized in terms of Child-Pugh A, Child-Pugh B, and Child-Pugh C in terms of the severity. So here is a distribution of the abundance of, for example, CYP3A4 metabolizing enzyme, and compared with control population, and you can see that the percentage of CYP3A4 compared to the healthy individual reduces or goes down as the severity of liver cirrhosis goes on increasing. Similarly, there is a significant change to the cardiac output and the blood flow to the liver, because physiologically also in severe liver cirrhosis, what happens is that the body adapts in terms of creating a portosystemic shunt in order to relieve the stress on the liver, and hence creates a bypass from the portal vein directly to the systemic circulation, bypassing the liver. Similarly, needless to say, the liver volume reduces as the severity of liver cirrhosis increases, and there is also a significant difference in the plasma albumin between the control and the various stages of liver cirrhosis. Similarly, there is a change in the renal function. Renal function tends to decrease as the severity of liver cirrhosis increases. And to a certain extent, there is an effect on the gastric emptying as well. And then there is also this formation of ascites, or the accumulation of water in the body cavity due to liver cirrhosis. So all these parameters are actually taken into consideration when you're generating a liver cirrhotic population. Similarly, we'll look into the Chinese population and how SimSip handles the Chinese population as part of the ethnic populations that we have. So we are looking at basically the liver volume here, wherein the liver size, it tends to be different in terms of when you compare it with the Caucasian, or as we call it, the normal healthy North European Caucasian population. The liver size is significantly smaller. So here we have compared the liver size with Japanese, because Japanese population is also another library that we have in SimSip. Similarly, there is a difference in the CYP abundance. So these are the variations in the various CYP enzymes, such as 2C9, 3A4, 3A5, 2C19, and 2D6 between the Chinese and the Caucasian populations. The blue bars are the Chinese population, and the purple or the violet bars are the Caucasian population. Also, there is an inherent difference between in the plasma proteins when you're looking at the Caucasians versus the Chinese population, and the distribution of phenotypes between the Chinese and the Caucasian populations for enzymes such as 2C9, 2D6, 2C19, and 3A5 is also very, very significant. So when there are these inherent differences in prime drug metabolizing enzymes, you cannot expect that the drug disposition would be similar between between the healthy or the North European Caucasian population versus any other ethnic populations. So this is just a performance verification on how the Chinese population in SimSip performs compared to the Caucasian populations, and this data is basically published in the reference year by Barter in 2013. So we briefly looked into generating the right kind of population. So now we are looking into optimizing the right dose using these PBPK models. So in terms of what influences, what parameters influence in terms of optimizing the right dose. So we are looking at basically the vulnerability of the drug towards the drug metabolizing enzymes, the transporters, the presence of phenotypes in your population, and the extent of drug-drug interactions. So when you are looking at optimizing the dose, you basically need to know the concentration of your drug at the site of action. So you're seeing what you're seeing here is the general PBPK model, and as I mentioned when I was explaining what the PBPK model is, there are certain compartments which are with a thick border. So and that basically represents some of the tissues which are very significant or which are important in terms of the disposition of the drug. So first we'll be looking at the liver tissue, or liver as the main site of drug metabolism. And in SimSip, basically we have the liver as a permeability limited organ, wherein we model the liver into various compartments, such as the extracellular water compartment, the intracellular water compartments, the sinusoids, and the canalicular membrane. So this basically represents the presence of transporters at these membrane boundaries, so that the disposition or the passage of the drug from one compartment to another is explained correctly. So we have these sinusoidal transporters, which basically uptake or efflux transporters, which act on the drug, and we also have the canalicular transporters, which basically pick up the drug and transfer it into the bile canalically, etc. So such modeling is important in order to know what is the right concentration of the drug at the site of action, or in terms of DDI, how much of the drug is a substrate to a particular enzyme, or is a substrate to a particular transporter, etc. Similarly to the permeability limited liver model, we also have the permeability limited gut model, wherein basically, which is the ADAM model, wherein we basically model the disposition or the transfer of the drug right from administration as a tablet into the way it disintegrates and then dissolves, partitions into the bile, permeates into the enterocyte. Whether is it acted upon after permeation into the enterocyte, is it acted upon by uptake or efflux transporters, and then we also model the concentration of the drug in the enterocyte itself. So basically, you're looking at each and every step of the drug right from administration to its presence into the systemic circulation or in the portal vein for the gut. Similarly, we have the mechanistic kidney model here, wherein it basically tracks the passage of the drug through each of the kidney compartments. And then also we have a permeability limited brain model in order to look into the effect of transporters which keep most of the drugs out or prevent them from entering the blood-brain barrier. And recently we have also added the permeability limited lung model, wherein you're looking at the pathway of the drug or the disposition of the drug in every small compartment of the lung from the trachea to the alveoli. Another aspect towards estimating the disposition of the drug in a population is the phenotypic status of the population. So basically, in SimSip, you can assign extensive metabolizers, poor metabolizers, intermediate metabolizers, or ultra metabolizers for CYP enzymes in your population. And similarly, you have the UGT phenotypes for your population, and also you can assign transporter phenotypes for your population. So these all aspects are important in terms of knowing what kind of metabolism or what kind of drug-drug interactions are you looking at when you're administering a particular dose to a particular subset of the population. So after looking at the generation of the right kind of population and what kind of properties do you need to look into in order to look at the right dose, we'll look at certain present and future applications. So here we have an example of eliglustat. So I had mentioned about eliglustat in my first slide when the FDA classified eliglustat as a personalized medication, because based on the labeling information, the dose would needs to be adjusted as per the phenotypic status, of CYP2D6 and 3A4 in the population or the patient in which the drug is being administered. So needless to say, eliglustat is a substrate for 2D6 and 3A4, and basically what you're looking at is that you need to monitor the exposure of eliglustat in CYP2D6 EMs and for metabolizers. And if you look at the labeling information, or if you look at the clinical pharmacology, the ClinPharm review from the FDA, you will see that it has mentions about PBPK simulations which were used for dosing recommendations. So basically, the label basically recommends that when Serdelga or eliglustat is administered with CYP2D6 inhibitors or CYP3A inhibitors, or CYP2D6 and CYP3A inhibitors, there is dosing adjustment required in order to not see any kind of overexposure or adverse effects of eliglustat in the patients. So in an overall, basically 12 cases were predicted in CYP2D6 EMs, IMs, and PMs in terms of the dosing recommendations. And these are the dosing recommendations. So you can see here that the usual dose recommended is 84 milligrams once daily, but the ones that are highlighted in green are basically the dosing recommendations derived from a clinical study. But the ones that are highlighted in yellow are basically dosing recommendations that were concluded from doing PBPK simulations. So looking at the assigning or looking at the effect of the drug in various CYP2D6 and CYP3A phenotypic populations. So you can see here, for example, that if a patient is co-administered a moderate CYP2D6 inhibitor such as terbinafine, then it is recommended that 84 milligrams once a day. But if a patient is given the Serdelga with CYP2D6 and 3A both CYP2D6 and 3A inhibitors, then either in poor metabolizers the drug is not recommended or contraindicated in extensive metabolizers and intermediate metabolizers. And more information can be obtained from the references mentioned below on that slide. Similarly, we have another example of aripiprazole, or the brand name Certara, wherein basically again primary elimination of the drug is via CYP2D6 and 3A4. And based on simulations, you can see that if the drug is co-administered with a CYP3A4 inhibitor such as ketoconazole, or a CYP2D6 inhibitor such as quinidine, there is extensive or significant drug-drug interaction. And hence, the labeling basically recommends that along with, if the drug is administered along with CYP3A4 or CYP2D6 inhibitors, the drug needs to be reduced in order to prevent any adverse effects in the patient. So similar to these two, such as eliglustat and aripiprazole, there have been several other new drug approvals wherein PBPK simulations have either helped in in optimizing the right dose, or in studying or in waiving clinical DDI studies. And hence, there have been basically 10 fast-track breakthrough priority or accelerated approval studies, from which 12 were for oncology, 3 for pulmonary, 2 for antiviral, 4 for orphan drugs, 1 for gastrointestinal, and 1 for CNS application. So basically, almost 100 label claims were informed by PBPK, including various areas such as drug-drug interactions, absorption, ethnic bridging, and different formulation studies. So when you're looking at the future applications, so one of the main aspects here is that when we are looking at the phenotypic status of the various populations that we were looking at, or the 2D6 EMs, PMs, IMs, or CYP3A4 PMs and IMs, what we are dealing with here is that you need to know the absolute abundance of these enzymes in your particular population. So when we are looking at such kind of information, we have good information for some of the populations, but when you're looking into, because of the development of highly sensitive techniques such as LC-MS, which are applied in the field of proteomics, you can now, with high confidence and high accuracy, determine the absolute abundances of these enzymes and transporters. And some of the references are given here, wherein groups from Manchester, etc., have published on the absolute abundances of these enzymes and transporters. So that's one of the future. So if such kind of data is generated even more for many populations, then these can be fed into PBPK models in order to have more accurate predictions of drug disposition. Similarly, there have been papers which discuss about use of RNA levels as a surrogate for the levels of enzymes in drug metabolizing tissues such as liver, etc. So if you basically determine the levels of blood circulating mRNA, these have a possibility of correlating to a biomarker for a disease and can be fed into such models in order to have more accurate predictions coming down to individual patient levels rather than just population levels. Another future application is basically generating the virtual twin. So generating data such as age, weight, height, sex, race, etc. can be achieved by mass data collection from particular patients. But the current rate limiting step or the current limitation towards making these models more accurate is basically obtaining data on metabolic enzymes and the genotypic status of the patient or population or the individual patient. Hence, you require a non-invasive method by which you can actually determine the levels of these metabolic enzymes and transporters in a particular patient rather than just a population. Because currently, whatever methods are available are basically invasive methods wherein a biopsy of a certain tissue or an organ is done in order to determine the abundances of these enzymes and transporters. Also, the effect of various environmental characteristics or genetic characteristics and what kind of effect does these have on the levels of these enzymes and transporters also needs to be known. There is a publication wherein SIMSIP has studied about the induction of CYP1A2 enzymes in the smoking population. But these are all based on population. You need to have this information on an individual basis, and we need to have a non-invasive accurate method in order to know the levels of this. So in a future scenario, what you're looking at is here that a patient along with his, like a futuristic scenario would be that you have a patient with his card which has all the information about the genome or whether he's an IM, EM or a PM for a certain enzyme. And he goes to a physician, and the physician basically has a handheld device wherein he feeds in all the patient information and the drug information, and the whole thing is uploaded onto a cloud, a PBPK PD predictor, which is based in the cloud, where the simulations are performed for a particular drug, and these are then again fed into the physician's handheld application in order to know what kind of drug disposition is that particular individual going to have for a particular drug. So this is basically the use of, as I mentioned in the very first slide, that though modeling and simulation and pharmacometrics, top-down or bottom-up, has been used in research and development, it has not been used at the point of treatment or at the physician level. So if the physician is able to get a handle on such modeling and simulation techniques, I think this will be it will have a great impact or a direct impact on the point of care or the point of treatment for a particular individual. So this is my final slide, wherein basically there is a healthcare summit which is being organized at the University of Manchester, which discusses more in depth about model-based precision dosing. And this event is held on the 19th and the 20th of May 2016, and which is sponsored by our organization, Certara Incorporated. So that was the last slide of my presentation. And so now, again, we go back to our poll question. So what do you think now? Can PBPK modeling and simulation actually help with precision dosing? If you can give your answers, we'll look at the poll results in just a few seconds. Oh, well, at least we have a very good response here. So earlier we had 66% saying yes. That has increased to 93%. And then we had a few people saying no, which has again reduced to 7%. And there was a major, oh, sorry, we didn't have anybody saying no. And we had 33% saying don't know, which has basically gone down to 0. So I hope my slides have conveyed some kind of information and helped these 33% to decide either yes or no. So I would really like to thank everybody for listening taking out the time and listening into my presentation.